Hello, everyone, and welcome to this Instruct webinar on structural applications of about COVID-19 that will be given by the Spanish Instruct Centre in Madrid, and that we will have today three speakers. It will be José María Carazo and Marta Martinez first, and they will explain about uh, the analysis of one of the proteins, the spike of the virus, and the flexibility of that spike. And then it will be me, and I will speak about how we got to those results and some methodological notes about structural analysis by cryo-electron microscopy in general. And finally, we will have José Ramón Macías that will explain about a portal that combines structural information with genomic and proteomic information. And this is 3D BioNodes that is also developed by our group. I will start presenting what is Instruct. So most of you probably are familiar with Instruct, but just a few slides just for letting you know. Okay, so Instruct is a European research infrastructure for structural biology research, and it is a kind of federation of research institutes and countries where they contribute with infrastructure for structural biology. And it supports 4 different kind of activities. We will see them in more detail, but they are research visits, training, internships, and R&D funding. So it is made of 15 members. So most of them are countries, but there are also international organizations like the EMBL. And it has 10 Instruct centers that are listed here. So in Belgium, Czech Republic, Spain, Finland, and so on. And 23 research sites. And it is mostly a European institution, but you have countries like Israel that is not fully European. And research visits allow researchers, not necessarily from structural biology, to have access to structural biology techniques. And there is a very wide catalog of techniques that are available, going from sample preparation, then biomolecular analysis, 3D structural analysis. And the idea is that the projects submitted to Instruct should be as integrative as possible, so requiring not just one specific technique, but a few of them, so that the synergy of having all of them and that the same umbrella is maximized. And the main activity is research visits. So research visits is you have a problem that you want to solve and related to the structure of a particular protein or particular complex, then you prepare a very short form and you register into Instruct, submit your form. The form or the application will be peer reviewed, and if it is approved, then you will have access to the facilities. So access to the facilities and the technologies. You will have access also to the consumables, the travel and accommodation, all for free. So it is paid through this Instruct Federation and or Instruct project. And then there are also training that it is very active in training in the different structural techniques, and particularly hands-on workshops that in which you learn how to use these techniques by yourselves. And there is a very wide catalog of different training activities. Then another activity are internships. So in internships, some people can go for one of these Instruct centers in Europe for 6 months to learn the technique in depth, and then come back to their host or home laboratory and apply these techniques more regularly. And there are also a program of small research projects. And these small research projects are primarily aimed at having preliminary results that later can be used for bigger fundings. And then, well, these are the two web pages. So Instruct-ERIC.eu. And if you want to keep up with the different news and everything, you can see there are almost 10,000 users currently registered. And one of the centers that the Instruct centers is ours. So it is the image processing center, and we are mostly focused on the image processing of cryo-EM data and also some soft X-rays tomography, but much less. So I think it is time for José María to start, so he can start with his presentation on the spike of the SARS-CoV-2.
First of all, thank you, Carlos Oscar, for the introduction, and thank you for the about 110 attendees that we have right now. Quite responsibility. So in the first presentation of these three series, I'm going to cover, with the help of Marta Martinez, on our work on the flexibility of the spike of SARS-CoV-2. Certainly, you all know SARS-CoV-2 as the virus causing the current pandemic, but what you might not know is actually these very nice tomograms that have just been published on the virus, in which the outer membrane is clear and a number of structures sitting on top of the outer membrane that are the spikes, the trimer of the S protein, that is what we are going to study, are there. They are there to interact with the cell, to recognize the cell receptor, and eventually to help fusing the membranes. This is a zoom up, and here you see the trimer. We are going to work with this specimen, with the trimer, which is the key to open the door of the cell and understand that probably more than 100 of the vaccines and candidate vaccines are really based on this system. So it's vital to know more about it. The pandemic came this year apparently from the blue, but not at all. We scientists in structural biology, we knew that things were really out there. What you see here are works from Dave Isler, 2016, Jason McClellan, 2017, in which other coronaviruses are studied here. MERS is a smaller pandemic that happened more regionally focused before CoV-2, and this is another type of beta coronavirus. The structure and even the flexibility that was already detected is the one that is happening in CoV-2, and now, because of these preliminary works or early pioneering works, we can now address better. It's a tribute to basic science and to be working always on focus. Well, in structural biology, we have quite a tradition of databases. In the case of cryo-electron microscopy, we have EMDB, and EMDB, as the beginning of November, had about 200 maps. From these 200 maps, the resolution of about 140 was good enough to make structural models. And okay, most of them were coming related to the spike for two reasons. One is because I was telling you is key for many of the therapeutical treatment that we have for the virus. And the other is that although in cryo-EM we can work with a wide range of specimen sizes, there are especially sweet spots. And specimen of about 500,000 Dalton with all the glycans and all that is in one of these sweet spots. So the important per se of the specimen to start with, and the fact that it was good for this technique explains a good part of this result. Of course, X-ray diffraction has a more processivity than X-ray at the time, and you see that they have produced a number of models. In their case, not only of the spike, but certainly of other proteins, and we have to acknowledge the great work of XChem, of fragment-based screening at Diamond, in which they studied thousands of compounds to see the interference with mainly the main protease and delicase. Great work. And you see that other technologies have had more limited scope so far, but I'm pretty sure that things will be changing very fast. But this is the picture as today. So we have a good specimen for cryo-EM, important, that we want to study, and we are going to do what everybody does. Let's go to classification and let's get different confirmation. Fantastic. However, please, my initial training was as a theoretical physicist, so I love to talk about first principle. If you go to our original paper, in which we described one of the most popular methods, the maximum likelihood approach for 3D classification in 2007, we were at that time making very clear the hypothesis underlying the method, and this was that all images were really coming from defined concrete structural classes. So we were not having the case of a continuous flexibility in the macromolecule, but we were having the case of discrete states that were the classes that we were going to classify through these procedures. Okay, we were not sure that this was really the case with CoV-2. So we work on really quite well. We worked to start with this is also a tribute of sharing data always and during this period, in collaboration with Jason McClellan. These are the images that his team took in Texas, and reanalyzing them through pipelines, image processing pipeline that I will not get into them. Carlos will get into them, but they were quite complex because we wanted to really get very good results. We were obsessed by quality, if you want. We should, I mean, in a system, always, but in a system that has so huge biomedical implications. Let me just focus in analyzing the flexibility. So at this moment of the workflow, we had a very good image data set. We had an ensemble volume, ensemble map, and then we did what everybody would have done. Let's do, for instance, rely on 3D classification. First warning, when we did two classifications, well, about 30–40% of the images didn't come out in a consistent classes or with consistent angular estimations. So this was a first warning that the specimen, something was perhaps not as simple as it looked at. And probably the non-simplicity was coming from the lack of discrete classes. And here, at that stage, what we say, okay, let's go for methods that do not require this discrete confirmation. Let's go for principal component analysis, which is a least square approach to funding this confirmation. But anyway, please let me show you our results with the consensus reconstruction. Let me just comment how good it is. I mean, how the working together on data indeed can improve the information that you get from them. This is, for instance, how a helix is coming in our map. This is how it was in the previous map from the same images. But on more qualitative grounds, here you have the local resolution that we calculate from the ensemble map. You see that it has many areas of high resolution. Of course, where there is flexibility, there is flexibility. And when we compare it with the previous map with the same images, we see that we have improved in local resolution. Not only in local resolution, I mean, there are more subtle concepts related to angular alignment. This is the issue of tangential resolution that you can find from Villas, José Luis Villas and collaborators. Here, what we did was, okay, imagine that we have, for instance, a rod that we had an alpha helix in the structure. And we are calculating rotations for angular alignment around, for instance, this point. Okay, we have an error in angular alignment. There are going to be rotations, and these rotations are going to create a problem of blurring that is dependent on the radius, and this is going to be especially noticeable in the direction perpendicular to the radius, that is the tangential resolution. And you see how the tangential resolution was very good in our case, was not so good in previous cases. And well, we went into the classification, and here you see the two classes with very good sharpening that you can find from the group in the CPM. And now let me present you what most people would be presenting you at this stage. That is actually, I have two classes, and I am doing a morphing between the classes, and you may think that what you are seeing is something dynamic, and that really I have information about all these intermediate steps. And this is not the case. I only have two conformations, and I am, in a way, inventing the rest. What about if I don't do that? What about if I calculate what is in between? And what is in between, I can access that through principal component analysis. Principal component analysis is a way to find directions of variability in my data set, finding those changes, those structural changes that really tell me more about the variance in the 3D map. Well, by this analysis, we could conclude that there were three multidimensional axes of variance, and when we see how the different conformations really occur along these three axes, we see that they are very isotropic. We don't have any suggestion that we have classes. And furthermore, when we project on this principal component analysis axis the closed and open form that we obtain through classification, we see that they expand very nicely axis 2, axis 1, but they do not say anything about axis 2 or certainly axis 3. So by classification, we are picking up the most, but we are not really able to tell about more subtle issues related to the other factors. For instance, here we have principal component analysis 1, and here is very clear that the movement of let me go again. You see that the movement that is happening, the closing is very clear. When we go to other components, we see that it's not exactly an opening and close, but it's a complete remodeling of the ribose receptor binding domain. And the case of factor 3 is exactly the same. Here, I would pass the presentation to Marta, that will be talking about map to model.
Good morning. Good morning. As you know, model building is the process that allows to get the atomic interpretation of the density map. And then the first step in this process is to improve or optimize the map to maximize the details and connectivity in order to favor the tracing of the atomic structure. We performed this map optimization using two different sharpening methods that we have developed in our lab, which are local blur and deep enhancer. And you can see here an example of the sharpening of one of our maps obtained with the deep enhancer methods.
You see that the map was done with a lot of care, but certainly there were areas in which we just did flexible fitting, like the ribosome, the receptor binding domain. And something that was very interesting is that we could see in our maps much better the glycans, and we extended actually the sugar exchange of these glycans. What you have here is actually a very quantitative approach to map to model fitting, in which we calculate by number of scores, Q-score, and Fourier rate correlation Q. That is a new thing that we are just publishing. And it is very good to tell us which area of the map are supported or not by a proposed model. in a quantitative manner that is directly coming from the information that you have in the half maps. So through very quantitative manners, we were able to improve before the map and then the model of these data that were being reanalyzed.
I don't want to finish this presentation without remarking that one of the main improvements of our model was to increase the extension of the glycan chains. The glycan chains are sugar chains that decorate the surface of the spike, and these sugar chains are quite common in most of the enveloped viruses that try to pass unnoticed to the host immune system. And we know that in our case, each protomer of the spike contains 22 glycosylation sites or sequence, and in this case you can see in the protomer A depicted 16 of these glycosylation sites. Compared with the previously published structure, we were unable to find additional glycosylation sites, but instead we were able to extend the length of these glycan chains that were already known.
Thank you, Marta. So just finishing, really talking on infrastructures, we have this history of a tradition of databases, and we have one to have the raw data of our analysis so other people could work on them, as we have done with JSON micrographs. And here you have, well, the screenshot of what you get in Empire. And still we have a long way to go, because this is really the data set with raw data, with micrographs and all that, that have been deposited in Empire as compared to these that have been deposited in EMDB. So still we have a long way to go. And this, just the last slide, almost out of time, and just to thank everybody. It's a typical slide of the COVID times in which we just join forces with groups, the best groups doing different tasks, and we really work the most to get the results of high quality and fast. And thank you very much for being here with us today, and we are open to questions.
Thank you, José María and Marta. There is a question on what is the difference between both sharpening methods and when it is suggested to use each one of them?
Okay, very interesting. I will answer, and Marta can also say things. Is that we work with the two of them at the same time while modeling, and there is not a way to know in which areas things are going to be better with one or the other. So we normally consider the sharpening as an enhanced view, not as a final result, but an enhanced view for modeling. And we use everything we can. But Marta, you did it hands-on, so.
Well, in terms of details, I think that the most recommended is local deblur. And in terms of connectivity, if you want to trace as better as possible the skeleton of the protein, I recommend the deep enhancer method.
There's another question. On which microscope was the data collected?
Well, it was in a microscope in Jason McLellan University, and it was a cryos with a K2.
Is there a danger of over-sharpening?
Yes, of course, there is a danger of doing anything. And for that, we use normally different sharpening. And also, always, we use quality control. So really, when I was saying before that we were calculating the informational content that a model had that was not supported by the map, this was not using any sharpening. This was using mathematical and statistical principles that were able to measure that difference, and was able to tell us, well, in this part of the map, simply you do not have, no matter what sharpening or over sharpening or whatever you have done, simply you do not have resolution. And we do that without the sharpening approach.
And is the EM tracer inside Scipion? I think you're mixing two things. One is
Yes.
deep tracer, the other one is deep enhancer. So the
Yes.
enhancer is inside Scipion, the deep tracer is not.
Yes.
Deep tracer is about modeling from ab initio modeling, so giving the map
Okay.
and
Very
the
good work.
sequence.
Yes, everything is accessible within Scipion, and if you have any doubt while using the software, there is email and a Slack channel to contact with the developers and to ask for any question. We would be happy to know more about your needs and to help you in getting your results.
Final question. Maybe you want to comment on the spread of continuous analysis methods for continuous heterogeneity.
Yes, absolutely. This is sort of the next frontier, because when we consider this constraint or this hypothesis in classification, we did it because we didn't know what to do if we didn't put it. But we all know that this is something to be improved. And at this moment, we have this principal component analysis that was first, the maximum likelihood principal component analysis that was first developed by him and Tagare and has been refined specifically for this project. It is a version of it is also in CryoSpark, and there are other ways to perhaps more powerful to approach this continuous flexibility. The work of Joachim Frank in Colombia is certainly paramount there, and you have seen his results on the ribosome and in other systems. And we are also developing other approaches on our own. Still, none of these approaches is widely used in the community because probably they need still quite some development. But clearly, principal component analysis that is the simplest, this that allow for continuous flexibility, is out there. It can be used, and in situations in which you do not have classes, and therefore anything you do is going to be unstable and, within quotation, beyond specifications, it is highly recommended.
Okay, thank you. So then I think we can move on to the next talk. That is mine actually. So I chose this title to be provocative. So what is behind the scenes? So we have been seeing with José María the results we've had with SARS-CoV-2 spike protein, and let's see how we got to it. And the main message would be one single algorithm is not enough to make it perfect. It doesn't mean that we made it perfect, but we tried our best. And in general, this is a very general message to the community. So if you just run one algorithm, you are bound to believe what that algorithm is saying. If you cannot compare to something else, you can never be sure that what that algorithm is saying is the correct answer. Still, if two algorithms say the same thing, you can never be sure either, but at least you are more sure. Okay, so these are the results of open and closed states that José María has been showing. And in cryo-EM in general, this is the kind of raw data we collect. So they are extremely noisy, very low contrast, and with very complicated mathematics. These are Poisson counts, and only when you start processing the raw data, you get something meaningful out of these acquisitions. And the first intermediate state is the one we are most used to, that are the micrographs. So when you align the frames, these images here, they're called frames. They're acquired at a very high speed. And when you align these frames into an image, then you start seeing the particles. And so each one of these circles is meant to be one of the particles contributing to the final structure. So all this process of going from the raw data to this intermediate data and then to the final structure, mathematically, it's all about estimating parameters. So when you estimate parameters, you can be in any one of these situations. So you have your observations, those are the blue points, and the red line here is the true model that generated those points. So in our case, the blue points will be the images, the model is the structure, and then you might have a very, very simple model that is this blue line here that tries to represent the true underlying model. And if your model has very few parameters, then probably what you have is a bias. And then on the other side, if your model has a lot of parameters, probably you adapt very well to your observations, but probably this is not a good model either, because it is not generalizing enough to other possible observations. So all this is about parameter estimation. So what are the parameters of your model? The parameters for us are all the parameters for the intermediate states. So you have to align your frames, you have to decide which are the particles, each particle from where it is seen. And then in the at the end, what you will have is a full model that is the map that is compatible with your images. And what we have recently observed is that normally we think of overfitting and over sharpening and all these things like a problem of having a lot of variance in our parameters. What we are seeing is that actually we are working on the other regime. So we have much more observations than parameters to fit. So whatever problem we are having in our maps is not because of variance in the map, but it is because bias in the map. So the thing is to identify sources of bias and try to minimize them. And for doing that, what you need is to validate all your intermediate steps. And so let's start with the first one, which is frame alignment. And frame alignment is the process of taking the raw data that looks like this and then finding either global shifts between frames or even local deformations between frames so that you can produce a micrograph where all the particles have been correctly aligned. And if you just use one algorithm, you will have one of these results and you will believe that is the trajectory of that movie. But the problem is that with different parameters or different algorithms, you might have different trajectories. So here, for instance, we are comparing 4 different runs with different parameters, different algorithms. 3 of them coincide, 1 of them does not because of a wrong choice of parameters. But unless you have the tool to compare the results of different algorithms, you are bound to believe that whatever you have done is just the right answer. And not only that, but different algorithms have their impact on the final properties of your data. So for instance, if you analyze what is the spectrum of a micrograph, and you might see that this motion core 2 algorithm, it produces micrographs with very high with a lot of energy at high frequency, while the rest they produce much less energy. And you tend to believe one of these more than this one. And it is not a criticism to motion core 2 applied to anything. So only that, again, you need for every specific case, you need to compare the output of several algorithms to be sure that what you are saying is the right thing. And even it has these things have their impact in terms of histograms, in terms of the local properties of the gray values and so on. The next thing is to do a CTF detection. So CTF is estimating the contrast transfer function of the microscope, estimating the aberrations of the microscope at the time of acquiring the image. And typically they look like this. So you have the power spectrum and then you fit a model to it and they look reasonable. You know that if you want to have very high resolution from theory, from first principles, as José María was saying, you need a very, very accurate determination of the defocus. And for instance, for a 2 angstrom resolution, you want to have an error in the defocus that is less than 100 angstroms, which is a very extremely high precision. And actually, this is what we observe in this data set. So when you just use a standard CTF detection, this is what we were getting. When you use several algorithms like GCTF, CTF find, and then XMIP CTF for the envelope, and you ask that all these algorithms agree up to 2.1 angstrom resolution in their prediction of the CTF, then it is only then that you get high resolution on your structure. So the errors that you commit at the CTF at the defocus estimation, they affect very much the final result. And normally we tend to look at the resolution of the micrographs independently, and resolution, you can think of it as having energy. So up to which resolution I have energy. So in this data set, we had energy up to 2 angstroms or 3 angstrom resolution, but it is not only energy, but this energy has to be coherent in all directions, and that is captured by astigmatism. And again, different programs compute different features of the images, and then you need something else. Resolution is reported by all CTF programs, but the non-astigmatic validity is only calculated by XMIP, and I'm sure that there are other features that you could look at your micrographs to make sure that the raw data is correct. So for instance, here we see that this particular acquisition, the non-astigmatic validity compromises really the highest resolution we can achieve. Then you need to do the perfect particle selection. So you need to choose which is a particle and which is not. So if you just use any algorithm in any way, this was in this particular case was XMIP particle picking, but with a bad training, and then you get all the particles, but you get many other bad things. And again, if you just use one algorithm, this is you don't have the power to realize what is a particle and what is not. For instance, when you use multiple pickers with more advanced algorithms like deep consensus, micrograph cleaner, and so on, then now you concentrate much better on what is a particle and what is not. Still, you have problems with the aperture. In this acquisition had a problem with aperture, and still the algorithms found particles in this area of the aperture, but this was the best we could do with the tools we had. And then typically you get rid of the contaminants in a 2D classification. So in a 2D classification, we use here CryoSpark, for instance, and then you identify good classes, you identify bad classes. So these are particles that really, or coordinates that were not really pointing to a particle, but you had also these classes with the aperture problem. So we came out with 300 coordinates. You have also like a small tools like remove duplicates or extract in a larger box, and then you're reducing a bit your number of particles. But out of this step, you would think, okay, I have used the 2D classification, and I think that most of my particles are correct. And when you use a different algorithm for 2D classification, you see that this is not the case. So some of the particles that still had an aperture problem, they were put by the previous algorithm in one of the good classes. So again, the same message. If you don't use a few algorithms, then you are bound to believe that the answer is always the correct one. And 3D classification can be another way of cleaning your particles. So for instance, we performed at the end of the previous steps, we had like 280,000 particles, and you use a single 3D classification step, and then you classify into 2 classes. You still see that 20% of the particles went to bad class, so they couldn't reconstruct a meaningful structure. This is very typical. And for the 80%, you would go further. But if you repeat this process, 3D classification is a relatively unstable process, and you discover that 10% more of these particles that were assigned to this class, supposedly to be good, they were jumping in and out of the good class, and sometimes they were assigned to the bad class. So you may think that these are particles that, because of the good class has a higher signal to noise ratio, they are attracted by that. We will go more into that idea later. Another idea is that you need to do a perfect angular assignment. The perfect angular assignment starts with a good initial volume. We know that this Einstein from noise problem that we are all aware in our field is a problem of the initial volume. If you have a bad initial volume, probably you will only recover that bad initial volume again. And again, you need several algorithms to try to get away from local minima, and if possible, a way to combine the outputs of all these algorithms in an automatic way, so that you don't bias the selection of the initial volume by a user decision choice. And then we are probably familiar with this kind of angular assignments in which some of the directions are overloaded, and you never know if it is really a property of the acquisition that you have done, so the sample lies in that way, or it is a problem induced by the algorithm. And that concern comes because we have seen very often, and it was proved in 2D, that when one of the classes, and one of the classes could be a 2D class, could be a 3D class, could be a direction in the 3D reconstruction, if it starts getting signal to noise ratio, then it attracts many particles around, and then the rest of the classes are empty. And this is one of the reasons, it is not the only reason, but it is one of the reasons why very often we get these biased angular assignments. And then if you compare, for instance, what are the angles assigned by one algorithm like XMIP high-res or rely on auto-refine, you see that for half of, depends on the data set, but from 10 to 50% of the particles, they get significantly different angular assignment and shift. And it is not only between two different algorithms, but also between two different runs of the same algorithm, because there are some random steps, especially at the initialization, and then the optimization pathways may be different, and you see that from 10 to 40% of the particles can have a different assignment, and you don't know if those angles are correct, the one for which they coincide. But what you are sure is that one of the two for this other half has to be wrong, and then those wrong assignments are contaminating your 3D reconstruction. The same happens if you use just one cost function to or one similarity function to compare your experimental images to your reprojections. So in one single similarity function, things may look like a single population, but if you look now with the different mathematics, now you realize that you have two different populations. And now you can start thinking these are badly assigned images, or they are images with a different conformation, or you might start to see what is the reason behind these two populations. But unless you have two algorithms, you can never realize that these things exist. And then we corrected for the frame alignment and CTF correction at the beginning, but or CTF detection. But it is very typical that you have to do it at the end, because you have more information and you can do a better job. But again, these algorithms, these are the Bayesian polishing and all this stuff. But these algorithms at the end, they are also very, very unstable, because you are trying to fine tune some parameters, and you are having a lot of noise, and then the estimation of these parameters is rather difficult. And again, you need several algorithms to compare the estimates of each one of them. And the same happens with the normalization. So each image has to be normalized independently, and sometimes you don't do it perfectly. And those lack of normalization per image, also that happens per frequency. So at the end, it results in some of the areas of the volumes being blurred or losing some small details, and that is where the different sharpening algorithms comes in. And you can compare the outputs of the different algorithms and try to decide which one of them is more correct. At the moment, we don't have any way to combine automatically the output of different algorithms. We're working on it, but so probably very soon you will have this solution also automatic. And then once you have tried to do your best, how well you did it. Okay, so the standard way is the FSC, but the FSC has two important problems. One is that it has a strong dependence on the mask that you use to evaluate the FSC. The other one is that the formulation of the FSC is rather good for random noise, but not for systematic bias. So if you have a bias problem and misestimating any of the parameters in between, the whole processing is a bias, then the bias is rewarded by the FSC. As long as you commit the same kind of errors in both halves, these both halves will be very consistent to each other, which is that is what the Fourier correlation is measuring. So you need alternative algorithms to tell you what is the quality of your final reconstruction. And you have all the local resolution tools, the directional resolution tools, and then different tools to compare your map to the model. And as a final message, I would like to highlight again this idea that the whole process is about parameter estimation in an environment where there is a lot of noise. And so how can you be sure that the parameters you are estimating are the right ones? You can never be absolutely sure, but what you can at least is to run different algorithms and make sure that they are all consistent to each other. And unless you do that, you are having a risk of making a biased result and having something that is not really the underlying signal, but there is something that is close to the underlying signal. And I would like to finish by thanking the people working behind all these algorithms. And then now I'm open to your questions.
A question that was how to interpret the bad particles. What do they represent? Just contamination?
Can be many things. So there are many possibilities. One is contamination. The other one is particles in different conformations. The other one is very often algorithms tend to pick coordinates that are off the center of the particle. So they are like significantly shifted from the center. And some other times they become attracted by some local maximum of whatever criterion they are maximizing. So there can be many possibilities. The carbon is particularly bad at attracting or making algorithms think that there are particles there. And then algorithms like the micrographium cleaner is really useful there. I would say that there is a trend now to try to pick everything and then clean in the 2D and 3D. I find that trend good. And you shouldn't use just one algorithm and then pick everything, but try several of them and then use something like the DM consensus, that the deep consensus that looks at the commonalities of the different coordinates.
You have a question that is how to make a good initial model if you do not know the structure? Yeah.
So a few years ago, that was really a problem. At present, there are so many algorithms that normally at least one of them can produce a correct answer. And then you need some way of choosing which is the correct answer. And for that, things like the swarm consensus of XMIP that is available in Champion, or there is another algorithm by Javier Vargas group. They normally are able to pick out of the many wrong answers, identifying which is the correct one. So it doesn't mean that they will never fail, but failures are much less common. If there are no other questions, I think we can move to the next talk that is by José Ramón.
Well, hello everyone, and welcome. So thank you for letting me the opportunity to introduce you 3D BioNodes. What is 3D BioNodes? It's a web platform that integrates a set of web services, meaning a set of tools that are available through a programmatic interface, together with an interactive 3D viewer to provide a unified environment in which biological annotations, mainly proteomics and genomics, can be interactively studied in their structural context. We have some tools that are mainly available through this simple programmatic interface, which is called API, and makes this kind of a standard web services. But many others are like the one to allow to calculate protein-protein interaction networks can also be accessed from separate sections of the website. So our users mainly provide just the identifier of the PDB or the EMDB entry that they are interested in, and they can show this kind of information in the viewer. But also they can upload their own structural models that they are working locally, for example, together with some own annotations, and they can analyze it in the same way as the regular entries in the databases. But yeah, many of our users come directly from the links that are provided by these main data sources or data banks, like the electron microscopy database. And yeah, probably the main component of 3D BioNodes platform is the 3D viewer. Here, the structures can be studied in a fully interactive 3D display at the left-hand side panel, together with all the relevant annotations that we can see in the right-hand panel. There are different types of annotations, which are organized in separate tracks with boxes and other symbols representing each annotation along the axis of the sequence. When clicking in one of these boxes or symbols representing an annotation, all the corresponding residues will be highlighted in the protein sequence alignment at the bottom left, as well as the atomic structure, as you can see here. At the same time, those residues will also be highlighted with vertical yellow bars, so it is easier to locate them in relation with other annotation types. And this is particularly of interest when you are trying to see or analyze features that are possibly quite separate in the sequence, but after organization in the space, in the 3D space, they are close together, so they are of particular interest sometimes. Additionally, a small panel will pop up with more detailed information about the annotation, including links to the origin of the data source. With the COVID-19 outbreak, there have been a big effort to provide the structural data for this SARS-CoV-2 virus. As has been said, as José María told before, many cryo-EM studies, but also X-ray, NMR, etc. Has been published, and also even for those less representative proteins, even modeling and computational predictions have been published. So we have added a dedicated website within 3D BioNodes with the intention to collect and present in a single place all the structural information available for this virus. In this new site, apart from some interesting selected examples, we have added a simplified schema of the proteome of the virus that can be used as an index with links, direct links to the corresponding sections for every protein in the virus. There, for every protein, we have a panel with a brief description of the protein and its role in the virus life cycle. And these panels contain all known structures organized in 5 sections. In the first section, we have all the atomic models published in the Protein Data Bank, the PDB, including validation data provided by external sites like PDB or Isolde, if they are available. And here the images are links to show the corresponding entry in our 3D viewer, like we've seen before, but also provide additional details and external links to the original data. In the second section, the EMDB section, the volume maps from the electron microscopy database are also collected and can be analyzed in a similar way. For some structures that contain more than a singular protein, we have grouped them under an interaction section. This can be interactions with other proteins of the virus or with the host protein. Or it can be interactions with small molecules, with ligands, that could be have been identified in drug screening studies. And then for a few viral proteins, there is no experimental structural data yet. So we have collected some computational models and predictions that could be of interest for these less representative proteins. And for completeness, all related proteins that from other coronaviruses, for example, as had been said before, SARS-1, MERS, that are very related to this virus, are also collected and displayed in this related section. Well, for this particular edition, we are able to provide a new set of annotations that are specifically relevant to COVID-19. For instance, the functional mapping tracks that allows the user to locate the residues that very likely constitute protein-protein interaction binding sites, and also ligand binding sites. These mapped residues have been obtained by modeling based on protein sequence homology from other coronaviruses. So all of these interaction amino acids can are putative functional sites and that have to be validated experimentally. Also, the drug screening track displays the different binding sites, this time experimentally obtained at Diamond Synchrotron in the UK in the XChem fragment screening campaign against the main protease and the NSP3 macrodomain proteins of SARS-CoV-2. Here, the tracks, where the tracks is expanded, I mean, you can see one of the row per different ligand showing the location of the interacting residues. The ones that correspond to the ligand, which is actually displayed in the 3D viewer, are colored in red, while the other that could be also obtained in the same experiment are, for completeness, shown in gray. In another category, the variants track summarizes the genomic variants for SARS-CoV-2 viruses that have been sequenced and studied, that have been compiled at the China National Center of Bioinformation. And in this case, the different rows represent aggregated observed mutations at a certain position in the protein, but most of these variants are still uncharacterized, apart from the ones in yellow you can see there, which are affecting start and stop codons. But in the link to the external origin of the data at the China National Center for Bioinformation are more in-depth information, so you can go there for more details. And lastly, for those cases where there is a cryo-electron microscopy map, we show some validation measurements of its local resolution. This way, it is quite easy to find interesting regions in the structure with similar resolution values and get a better understanding of the flexibility or conformational dynamics of a structure. Local resolution scores are calculated in the group, as has been mentioned before, with different algorithms, deep res, mono-res, so on. And if there is an atomic model available for that particular map coming from PDB, we can also calculate MapQ and FSCQ scores. The local resolution values in these cases are projected to the atoms on the atomic model using a convenient color code depending on different thresholds. For example, in MapQ, red indicates atoms with low resolvability, while for FSCQ, red indicates atoms that may be associated with noise, possible overfitting, and blue indicates poorly fitted atoms or areas with low resolvability. And that's all. I finally would like to also thank all the people at the team at the biocomputing unit for all the cooperative and hard work. And yeah, that's all. Thank you very much for all your attention. And I'm happy to take questions.
Well, there is a question that is, thanks, very interesting tool. Apart from SARS-CoV-2, how do you think it can be useful? I mean, who will enter all the information and who will be able to use it?
Well, I was explaining mainly the part that was dedicated to SARS-CoV-2, because this is the main topic right now. But 3D by nodes can display any entry from PDB or EMDB. So yeah, after the success, I may say, of this subsection in the website related to SARS-CoV-2, we are thinking on preparing different sections related to different topics, for example, cancer, or maybe some particular complexes that can be like a one-stop shop, say, for any particular group of users, that they can go there and find a list of all the relevant entries in the different databases related to a specific topic, for example, cancer or whatever. But yet, any entry in the different databases can be displayed. And as I mentioned at the beginning, even the local models, or the models that hasn't been submitted yet to the databases can also be sorry, load into the website and compare with all of the annotations that are available for that particular protein.
So what do you need to upload then? So you need to upload the map, the atomic model, and the sequence, or?
No, you just, well, the list, you need to upload a PDB file format, CIF or PDB, anyway. Then if you have the map, you can also upload the map, and we can perform a blast query about the sequence that is already in the PDB file, so we can find similar proteins that are already in the databases and retrieve automatically all of the annotations related to that sequence, so that possible similar proteins.
Information of 3D bionodes is accessible from the EMDB, right?
Yeah, yeah. I've been focused most in the interactive side of or part of the website, which is the 3D viewer. But as mentioned, also there's a big set of tools that provide all the annotations in a programmatic way. I mean, it can be retrieved by other web services. So yeah, it is accessible through these web services directly or from the EMDB or PTP. Yeah, we have links, direct links here. You see in the EM database data resource, which is the American part of the database, and also in the EMDB, which is the European side of the database.
Okay, so it seems that there is a question for me. Is the fact that you need to use different algorithms for each step mean that the algorithms don't treat noise correctly? Could there be a good algorithm that would treat? If you did, when you work with algorithms, you easily realize that algorithms are meant to estimate something. And if you are in a noise environment, it is not that they cannot or they are not dealing noise correctly. It is that because of the noise, they make mistakes. And this is something known in the theory. So when you estimate parameters in a noise environment, your estimate of the parameter is noisy itself. It is another random variable. And unless you have, so the way to fight the noise in the parameters is by doing several estimations of the same thing and averaging them. And then it is where either you run your algorithms a few times and take the average of all those, or you run ideally different algorithms with different mathematics behind, so that they have different ways to estimate the same parameter. But otherwise, because every time you estimate the parameter, you have noise and may also have bias. And then the way to identify the bias is by using another type of estimator, because otherwise, if you use the same type of estimator, you might be incurring twice into the same bias. Okay, so if there are no other questions, I think we can stop here. We are just on time. Thank you all for attending. It has been a pretty crowded seminar with 100 people. So thank you all, really.
Thank you.
I hope it has been useful.